All right, we're go. So in advance of class today, you had a little bit of reading on the tree thinking article, and you're going to take a self quiz. We're going to get into that. So today's agenda looks like this. First, I have a segment of herpetology in the news to share with you, a little bit about programmable frog cells, which is pretty cool. Then we're going to review that article you read for class today. And I have the answers to that quiz that you took. You're going to give yourself a grade. We're going to debrief that and make sure all those concepts are solid for you. Then I'm going to lecture a bit on amphibian diversity. So just kind of introduce you to the variety of amphibians with a focus on families that are that live here in Ohio. And then we're going to talk about how to read literature from a paper from the primary literature. Your assignment for Monday next week will be to read a paper on mud puppies, and I'll get to that. And then whatever time is left, we're going to watch a little bit of the Life in Cold Blood series. Some of you have probably seen this. It's pretty old at this point. It's over 10 years old, but it's brilliant. It has some great footage. And we're going to watch the episode on amphibians with thinking about some specific aspects of amphibian biology as we watch, and we'll wrap things up. So that's the plan for today. Before we dive in, what questions do you have for me about course logistics or what ways do for your program for today or anything like that?
I'm really asking for the guy ways to meet your, like, are you gonna test this on the use things?
Oh, right. No, I won't include questions on that. That's just like a bonus. It's just like free knowledge.
And then
I'll get there. You'll see what we're gonna do. We're gonna do an activity with it. Good question, Sid. Thanks for clarifying that stuff. Any others? All right, so herpetology news. So this isn't really herpetology. I agree, but I saw this recently. It kind of blew my mind a little bit. So they use the stem cells of a type of frog, the African clawed frog, Xenopus laevis, very commonly used organism in the lab. It's actually invasive throughout much of Europe. You guys, maybe you've seen them. They're really common in the pet trade as well. They're entirely aquatic. I should pull up a picture of one, but you've probably seen them before. Anyway, they figured out a way to make the stem cells of this frog do what they wanted. And so the quote, I love the quote here, they're neither a traditional robot nor a known species of animal. It's a new class of artifact, a living programmable organism. And I saw this, it kind of blew my mind. And I want to show you the video about this. Let me play this. I need to adjust the sound, so bear with me as I adjust that. Oh, there we go, it worked. Xenobots, that's what they call them. That's just an overview. I put a link to the original scientific paper as well as a news article on it in the program for Monday if you're interested. So I'll do this periodically, just new research that I think is relevant to class and you might be interested in. And that one just kind of blew my mind that they could basically make these cells. And actually, there's been an advance since this article came out, is they can actually make them so they self-replicate. So they have the cells in a certain structure. And when they find other cells that are loose, they'll gather them up and make them into that same structure. And then those ones can go and gather new cells and do it. It's freaking wild. So check it out. It's interesting. Anyone see this article when it was out? This one's a little bit older now. It's from last year, I think. Well, I'd say even from 2020. It's from a little bit older. So interesting stuff. Anyway, let's go back. Evolutionary relationships. So this was from last week. Probably looks familiar. I asked you about it at the beginning and end of class. Many more of you got this answer correct at the end of class than at the beginning. The correct answer is that they're both equally correct. Both of these phylogenies are exactly the same. And what probably tripped you up was the fact that they're in a different order. That one's fish, frog, lizard, mouse, human, and this one's fish, lizard, human, mouse, frog. Now, a very common misconception when we think of evolution is that evolution works like this. That evolution is like the step up process from less complicated to more complicated, or from like lower, quote unquote, lower forms of life to quote unquote higher forms of life. You notice very conveniently that we think about evolution that way humans are always at the top, like we're the pinnacle of evolution or whatever. That's completely incorrect. Evolution does not work that way at all. It's not a progression necessarily. It's not a progression in this way. What it is a progression through time. Remember that all organisms that are all species that are living today have been evolving for the same amount of time. We all share that same common eukaryotic ancestor from however many millions of years ago. And so we've all been evolving for the same amount of time. So it's really incorrect to say that one group is more evolved than another. What we can say sometimes is that some groups have more derived characteristics, which means they have more kind of specialties or special features, but it doesn't mean they've been evolving for longer or better. And so you see here, this is kind of like a misunderstood traditional view of evolution if you're reading it that way. But remember, you don't want to read it that way anyway. What you care about is these tips and how they are related. You want to look at that topology. And you notice here that these are exactly the same, only they've been rotated, specifically around this node. You flip this whole thing around and put the toad back over here, and then flip it at this node and put the lizard back on the other. No, actually, oh, excuse me, flip this here and flip the mouse and the human, then you can recreate, you can move from one to the other. And so these convey the exact same information. And if generally, if people are asked which is correct, they might be tempted to choose this one because it seems like it's this progression up towards the glorious organism that is humans. But in fact, this one is just as accurate. And there's other ways that you could change this as well, and they'd be just as accurate. So I just want to make sure we've got that. And this is an accurate representation of a very broad tetrapod family, well, tetrapod and fish is the outgroup phylogeny. Of course, it's a lot more complicated because there's a lot more stuff in there, but that is generally accurate. Questions on that? Anything lingering that's still not clicking for you? That's okay. Mm-hmm. That's it. That's a super good question. So the question is, let me repeat it back to make sure I understand, is that I said that all organisms have been evolving the same amount of time, but we talk about different rates of evolution, right? And so that's it. So the difference is, I'm trying to think of a good analogy. Okay, the difference is this. You and I are both going to move for 10 minutes, right? You're going to run, I'm going to walk. At the end of 10 minutes, we've both been traveling for 10 minutes. We've both been traveling for the same amount of time. However, you've been running, so your rate is faster. So you end up moving farther than me, right? Now, it doesn't mean that that analogy holds up in terms of the rate. But what's important to think about is that we don't have the same finish line. So even though you've been moving more, you maybe necessarily have been moving in other directions or anything. You've just, you've experienced more variation in that specific trait. And so we can talk about rates of evolution for specific traits. So for example, I have a good friend that studies the rates of evolution of thermal tolerance in lizards. And we know that generally speaking, cold tolerance evolves faster than warm tolerance, right? There's a bunch of reasons for that. It doesn't mean that those species have been evolving for different amounts of time or those species are differently evolved. It just means that one specific trait is evolving at a faster rate compared to others. Does that clear it up? That's a great question. I'm really glad you asked that. It's a complicated answer. Good. Other questions? Do you talk about rates of evolution in your evolution class? Those of you that have taken it here? Comparative in that sense? Okay. Yeah. And we can, what's important to note too, and just one more point, is that we can only talk about rates of evolution for the same trait. Like you can't compare how fast lizards evolve thermal tolerance and how quickly rodents evolve longer legs. Like those two things wouldn't be comparable. It would only be like the same trait that you can compare. Cool. Well, let's talk about this a little more. I want to kind of continue with this. The answers to the tree thinking quiz are now available in Blackboard. So you took a 10 question quiz in advance of class today. What I'd like you to do is go ahead and grab those answers off of Blackboard and review, compare that to your own answers, essentially grade your own quiz. And then as you're doing that, go ahead and complete this survey that's going to ask you how many questions you got right. And then, ask you to reflect on that a little more. This is worth 2 points for participation, but note that you're not being graded on how many you got right. You'll get the same grade if you got 10 right or if you got 0 right. What I'm doing here is checking for understanding and giving you a chance to make sure that see if you have any questions you need to ask or anything like that. So we'll give you 10 minutes to work on that individually. Princeton and I will come around and check in, answer any questions, and then we'll take things from there. Does that sound okay? Cool. I'm just going to step outside for 1 second. All right, so the idea here is I will take a look at your surveys. I'll see how everyone's feeling about these concepts. And then given that information, I'll kind of assess how much we want to get back into this again next week. So between the lecture yesterday and the reading and the practice, we've kind of seen this stuff from a lot of different angles. The quiz and the reading was tricky too, because it has phylogenies of like different shapes. It have some where all the lines are kind of at angles, and then other ones, they all convey the same information, even though they look differently. Although it seems to be more confusing sometimes if they have a certain shape. And that's why I want you to take that so you could see how the different ways that phylogenies might be depicted, even as they have the same information. So I'll get that information. We'll see what we want to cover with that next week. Before we move on to our next topic, do you have any questions? Yes, please, McKenna.
Is there a certain format that's more like preferable in a like professional environment? Is it like really variable, or is there like a particular one?
Awesome. It just depends on who's doing it, what they're trying to show, if they're trying to show species or traits or genes even. I've seen it a lot of different ways. And I'll say in all honesty, like my own research doesn't ever use phylogenies. Like I don't look at things in a comparative context and do that. So I don't have firsthand experience thinking about like, oh, what's the best format for a phylogeny for me to use? But I read a lot of papers with them and I've seen them all kinds of ways. So yeah, there's no standard way to do it, unfortunately. So it'd be easier if there were. But I think because the different formats are conducive to different ideas that you want to express. So yeah, that's a good question. Any others? All right, well, fasten your frog belts. Let's get on to amphibian diversity. I don't know what a frog belt is or where that even came from. Amphibian diversity. So we're going to talk about all of the cool amphibians in the world very broadly, and we'll focus on a handful of families here in Ohio. So as you saw in lecture on Monday, there's 3 major groups of amphibians. There's the salamanders, also known as Urodela or Caudata. There's about 7. These numbers are actually a little low, simply because we're adding new species every year. So there's probably 700-800 species of salamanders right now. We also have Sicilians, which are known as Gymnophiona or Apoda. Remember from your Latin that pod means foot, and so A means like there isn't one, so they have no feet. And remember that, like cephalopod, like your octopus and squid, cephala meaning head, pod meaning foot. They're like feet head because they just kind of are mushed together. Anyway, just those Greek roots will help you remember some of this stuff. And then frogs and toads are in the group Anura. There's only about 200 or so species of Sicilian, but there's 7,000 plus species of frogs and toads, right? Don't worry about these exact numbers. I'm not going to quiz you and say, how many species of salamander are there? what I want you to take away from this is that there's a lot of amphibians and most of them are Anura, right? And a good number of them are salamanders. That's the takeaway message here. The exact numbers aren't what's important, just the relative values. And so we go back to our phylogeny. This is a phylogeny of stuff we're studying in this class. This has this is a phylogeny of the major vertebrate clades here. I've got fish broken off. This actually isn't terribly accurate because fish are actually a paraphyletic group. And so we're actually nestled in a group with lungfish and some other kinds of fish. And so we're just as closely related to like a lungfish as a lungfish, say, is to a shark. But I just lumped all the fish together for simplicity here. But just keep that in mind. We're all just derived fish. So here we have our amphibians, right? And remember, amphibian is monophyletic. It's a true clade because it includes all of these groups from the most recent common ancestor and all of the descendants of that most recent common ancestor. So amphibians is a true clade. Notice that they're sister to the group that includes mammals and reptiles, including birds, right? When we say sister, we mean that all these amphibians are equally related to all of these because they all come back to this most recent common ancestor here. So in other words, you and I are just as closely related to a salamander as a lizard is to a salamander in an evolutionary sense, right? Because our most recent common ancestor with amphibians is back here, as is the most recent common ancestor of amphibians with snakes or what have you. Sound good so far? All right, so in amphibians, let's talk about salamanders first. Salamanders generally have 4 limbs and a tail, though there's some minor exceptions to that. They're segmented very often by what's called costal grooves. And I'll show you a diagram in a second. Costal is basically just refers to the ribs. And so you can see actually really nicely in this guy how their long body has all of these kind of grooves along it. And those are just, that's at where the ribs and the musculature around the ribs form those kind of ridges. They can be terrestrial or aquatic. Some species can be both depending on their life history phase, and they can go back and forth, but they can, if you can find these guys on land or in water. They don't have middle ear cavities, so they don't hear in the same way that other vertebrates, including us, do. And they exhibit both metamorphosis and direct development. And so I want to make sure that these terms are clear. Metamorphosis. Oh, this chalk sounds terrible. I wish I didn't have it in your ears right now. Metamorphosis. Metamorphosis means that you transition from a larvae into an adult at some point in your life history, right? It means at some point you're a larvae and then there's a big transition, like a, not just a getting bigger transition, but like a marked transition. So the great example of this is frogs. You've got your tadpoles and you've got your adult frog, right? Salamanders are the same. Some exhibit a larval form. So for example, mole salamanders. So for like tiger salamanders or marbled salamanders, when they're young, they're aquatic, they have gills, and at a certain point, they change, not in a second, but in a very short amount of time, they transition and they become terrestrial adults. So some salamanders exhibit metamorphosis. Others have direct development, which means they don't have a larval stage. They just have, when they're the babies, essentially, I'm going to say babies look like small adults. So they're obviously smaller and they need to grow, but there's no like marked difference in their body structure or anything like that. So for example, the Plethodon salamanders that we'll actually see later in the semester, the babies are born and they're just super teeny tiny little cute adults. They don't look any different. They don't have gills or anything like that. So we have both forms of life history in salamanders. Question, Chase, go ahead.
Isn't there a word for that that's not direct development?
Isn't there a word for that that's not direct development?
I feel like I learned it in entomology.
Yes, there probably is, but I can't think of it off the top of my head. There is another word for it in my, you all know entomology better than I do. What are the terms in entomology of how many instars they have or whatever, if they have direct, what's holotabulous versus? Yes, which one is when they don't have any, like, that's hemi,
Hemimetabolous is when you have like stars. Holometabolists is metamorphosis.
yes, okay, and they have metamorphosis. But there's no insects, there's no arthropods that have direct development though, right? They all have some kind of larval form or instars. Is that right? No? Okay, see, I don't know this stuff.
So that's the one.
Okay, that is we don't, yeah, so that would be the concept is the same, but we don't, you never see that word used for these guys, at least that I'm familiar with. But that's, yeah, it's a great connection. So, and fortunately for us, this is much less complicated than we see in insects or anything like that, because they have all kinds of crazy stuff going on. That clearly I'm not an expert in. Sid, go ahead.
For the coastal grooves, is that like akin to intercoastal for even in your ribs? Is it like the muscles, or is it just some sort of weird thing?
Yeah, so costal just refers to ribs, and so we have costal muscles, that the muscles that surround our rib cage and help us breathe when we're breathing heavily and things like that, or exhale or inhale and exhale. And the salamanders, they're called costal grooves because it's just along the ribs there. So they are homologous structures in the same way that like you have some grooves in your ribs. It's basically the same thing, but they have longer bodies.
So it's not muscle then, it's just
Those grooves, that's just an external structure formed by the rib cage and the musculature around it. Just like if you were to look at this part of your chest in the mirror, you'd see a bunch of lines of your ribs out. Exact same idea.
Those grooves that aid in the gas exchange, right?
But they can in some species. It's not necessarily the grooves though. They actually have, so take a look at our hellbender in the jar over here by Chloe. You can see he's got like really loose, flappy skin, and that is to increase the skin surface area for gas exchange because they're aquatic, right? And so they can actually breathe through their skin. The bigger surface area you have as their skin, the easier that gas exchange is to happen. The costal grooves don't really facilitate that because they're not really in, well, I guess Plethodons are terrestrial and breathe through their skin, but you don't really see that so much in aquatic species as in terrestrial. So good, that's good. These are great questions. Yeah, we do have the hellbender in the jar over there. He's pretty cool. You'll see pictures of him not in a jar in a second. All right, so salamanders can get big too. This is a Chinese giant salamander. We often think of these guys as little dinkers in the forest, at least I do, but they actually can get quite large and historically even much bigger than that. Historically meaning like millions of years ago before humans. So there's 5 families in this of salamander in Ohio, and I'm going to walk through each of these in a little bit of detail, but not too much detail. And those 5 families are Ambystomatidae, Proteidae, Salamandridae, Plethodontidae, and Cryptobranchidae, or Branchidae. I think it's a hard K here, Branchidae. Go with that. So we'll know those 5 families. You can list them now, but we'll go through each of them and give some examples here on upcoming slides. So the big takeaway here is I want you to know amphibian diversity generally, and we're going to focus on knowing some of the species that live here in our beautiful state of Ohio. I don't think I've ever said that before, actually. No offense, Ohio. I'm still new to Ohio, so I don't really know too much about it. Not much to know. It's, well, Cincinnati was cool. We had a lot of fun in Cincinnati. There's lizards everywhere. It's awesome. It's like paradise. All right, so just to take a quick look before we get into those families, I want to just give you some basic vocabulary when we talk about the anatomy of salamanders. So here's some diagrams. Here's those costal grooves. You can see how there's grooves all along the torso of the body. The head is anterior to the neck, not terribly surprising. An important measure that will come up again in this class is what's called snout vent length, or SVL. This is kind of the standardized way we measure the size of amphibians and reptiles. And that is the distance from the tip of the nose to the cloaca or the vent, right? And so it's basically the body length excluding the tail. And the reason for that is simply that a lot of these organisms can lose their tails or drop their tails. And so the total length doesn't really tell us important information because that tail length can be really variable. So the lizards that we study in the lab, for example, they can drop their tails. And so I might have 2 lizards of the same size. One has a tail that stops here. One has a tail that stops here. And so if I compared their size using total length, it'd be really misleading And so we standardize that and just do the snout to vent length because that's always going to be the same. And that's used for salamanders and it's not really used for turtles so much, but it's used for alligators, snakes, lizards, frogs, everything but turtles that we'll cover in this class. The term vent is synonymous with cloaca. There's another vocabulary for you. Actually, Princeton, would you mind erasing this and writing vent equals cloaca? Please and thank you. A cloaca, it actually comes from the Greek word for sewer. Back in the day, in cities, they didn't have anywhere to get rid of their waste. So there were literally these like gutters outside of houses, and they would just dump all their household waste and feces and all, you know, all their toilet waste and all their food waste into these open sewers along the streets. And so the streets really smelled bad, and that was called the cloaca. And so we use the same term for amphibians and reptiles. They basically have a single orifice through which they excrete nitrogenous waste, so their urates or urine, and as well as their feces, and as well as it's a place where a lot of either the intromittent organ for sexual intercourse is emitted or received, or sperm is released in the case of external fertilization. So it's where all the action takes place, right? They have one hole for everything. That's an easier way to say it. We also have a diagram here that shows you some of the coloration on a salamander. And I don't want to get into this in too much detail, but it's just some vocabulary of how we describe the colors of these guys. We have what's called ink spots, which are these kind of little dots along the belly here. Big spots, it's kind of self-explanatory. And then you see flecks of color. I won't get into that too much, but I just wanted to show it to you because you're going to see that come up when we look at some field guides and diagrams and things like that. I just noticed special thanks to Jeff Davis and Greg Lips. That's funny. Okay, so let's look at the first family of amphibians, salamanders in Ohio. That's Ambystomatidae. These are commonly called mole salamanders because they live underground. They are primarily terrestrial. An important indicator is that they have only 4 toes on their front feet and 5 on their back. That doesn't seem important now, but when we get to some other species and we want to be able to tell one from the other, that will become an important characteristic. They have 4 front and 5 hind toes, and they don't have this nasolabial groove, which I haven't defined yet. Is that on here? No. Okay, hold that thought about the nasolabial groove. I'll come back to that. It's basically this little groove between their eye, it's actually more between their nostril and their mouth, in that area anyway, where they often release pheromones associated with courtship. These guys don't have it. And you can see this is Ambystoma maculatum, the spotted salamander. We will hopefully later this semester take a trip on a nice rainy, warmish fall evening, probably sometime in early mid-March, down to a nature preserve south of town, and we'll see all of these guys getting out and getting ready to, or not getting ready to mating in the vernal pools. It's super fun. I've gone out the last 2 years and we've seen a bunch of them. We're out there at night with flashlights. The chorus frogs are like deafening, and you're trying to walk around without stepping on salamanders. You get all wet and cold. We'll bring some hot chocolate. It'll be great. So we're going to see these guys later this semester. So Ambystoma maculatum, that's one example of a salamander in the family Ambystomatidae from Ohio. Yes. Yeah, let's I think I have that on a slide. Could we hold that thought for a second? I think I have a better diagram on a slide up here, and if not, then I'll just draw it on the board for you. That is a great question, and we'll get to it, and don't worry about it right now. I'm not going to ask you a question about that on the quiz. I will ask you questions about families of salamanders in Ohio, though. The next family is Proteidae. These ones I'm super excited about because we're going to spend basically all of next week learning about this one species, the common mud puppy, Necturus maculosus. So we're going to go to the field site and see them. You've got some readings for Monday on these guys, and we're going to talk a ton about them. They're so cool. They're really big. They're beautiful. They're just awesome animals, okay? Importantly, the adults exhibit the same characteristics as the larvae, and this is known as being paedomorphic. Paedomorphic comes from the root for young or child, and morphic meaning like your form, morphology. And so they have the morphology of a baby, essentially, meaning that the adults don't lose their gills and they have the same body shape and the same aquatic lifestyle as larvae of other closer related species, even though they're adults. These guys have 4 toes in front and 4 on the back. And that's important because that's different. Remember, Ambystomatidae has 4 in the front and 5 in the back. And so the larvae of these guys, I don't have a picture, actually looks a lot like this. And so if you get one in a jar, do we have any of these guys in a jar around? Those are mud puppies, right? And then, yeah, I was just, okay, we don't have any. I thought maybe we had some larvae mole salamanders, but the larvae of these guys look similar to this. But if you count toes, you'd be able to tell the difference. So you count the toes on those guys, and you should say that they have 4 in front, 4 in back. They're entirely aquatic, but they do have lungs. They can air breathe as well. And they also exchange gas through their skin too. Yeah, go ahead, Sid. A juvenile and the same way you would tell apart a direct developing organism, and then that's simply by its size. And there's other external characters that can change. So mud puppies, I actually don't know the answer to that specifically, but others, for example, the lizards we study, when they become, the males become sexually mature, they change aspects of their color and they get these big pores, and you can see other kind of external things change. Kind of the same way humans, I guess, too, right? You can kind of tell. I have a 13-year-old son, so I'm really thinking about this transition a lot. Anyway, that's neither here nor there. So that's Proteidae. The other family, 2 more families, or 3 more families, actually, sorry, Salamanderidae. These are commonly called your newts. You see these are very common across the world, right? The species we have here is Nutophthalmus viridescens, the eastern newt, very common. This is a F stage. These guys have this really interesting life history where they actually start off as aquatic larvae, and then they transition and become these really brightly colored, what's called red Fs, and they're completely terrestrial. And then as sexually mature adults, they go back and live in the water and mate in the water. So they have like this 3-part life history where they're aquatic, terrestrial, and then aquatic again. They do not have gills as adults, even in the aquatic stage. They do have lungs. And they have this very kind of granular, especially in the F stage, this kind of granular skin. And they do release toxins. And so these guys produce toxins in the same family as like a pufferfish, tetrodotoxins. And it inhibits the functioning of your, essentially as the firing of the action potentials of your nerves. And so it can lead to paralysis or worse. So don't eat these or lick them. You won't get a buzz, you'll get a numb tongue. Yes, Ethan.
Would you say that the toxins were the defining characteristics
of this family? Good question. Is it defining characteristic of the family? Many, many species in this family have these toxins, but I don't know if all of them do. So
I think
some might have, they might not have it.
I don't, because they have the
I'm not 100% sure either. A lot of them do though from this family. So there's Tarika newts out west, are very common. Eski, I'll get you in 1 second. And they actually can be really quite toxic. There's the story about the hunters who, you know, the story that they were out, I don't know, camping and hunting one weekend, and they found them all, the 3 of them all dead at their campsite. And they had a coffee mug like on their fire. They were making coffee, and there were like a bunch of newts in there. And so apparently either the newts crawled into their coffee mug and then they drank the coffee and died, or they were like, sweet dude, let's trip on some newts. And they threw newts in their coffee and they tripped pretty hard. Did it? Yeah, it's crazy. It's, yeah, I don't know. So don't lick a newt. Okay, SK, go. Yeah, if you die, that's permanent. No, what would probably happen, in all honesty, these guys aren't super toxic like the ones at West. I'm not saying you should do this, but if one did, if a little kid did and didn't know any better, it was like, ooh, newt candy, you'd probably get a very numb mouth and lips and tongue for a while, and then it would probably pass. If you just like you didn't eat the whole thing or whatever. It would just inhibit any of those nerves from firing. And so it'd be like when you get back from the dentist and you can't feel anything and you can't move your lips the way you want. And it would be kind of like that. It would actually be remarkably similar to that. Now I think about it. I keep saying that. No, two more families. Plethodontidae is the other one. So these guys are super cool. They are the most diverse group of salamanders. And in fact, they are the majority of species of salamander fall into this one family. And what's cool about them, well, there's a lot of cool things about them, but they're lungless, which means that the majority of salamanders species are in fact lungless. So they're terrestrial, but they don't have lungs. All their gas exchange happens through their skin. So as you can imagine, they need to keep that skin nice and moist at all times. This is the redback salamander, Plethodon cinereus. This guy is found across all of the northeastern US into southeastern Canada, and they're super abundant. So in a lot of places, these guys represent the greatest amount of vertebrate biomass. In other words, if you took a plot of land in a forest in the Northeast and you picked up all of the vertebrates and you weighed them, the vertebrate with the most weight would be these guys, more than your bears, more than your deer, more than your raccoons, whatever. You'd have by mass, total mass, you'd have more salamanders than anything else, which is cool because we want more salamanders than anything else. So they can be really abundant. You probably, anyone who's taken a hike around anywhere around town here and flipped over a log, you've probably seen some of these guys. We study these guys in the lab. We have cover boards set up down at Krause and at another site out east of here. And in class, we will go, and I think we're going to have time to have a field trip down there and check out the cover boards and see that. We should talk about that. That'd be cool. So yeah, these guys, and there's a number of other species in this family that live in Ohio as well. I'm just giving you one example. All right, our last salamander family is Cryptobranchidae. These guys are kind of paedomorphic, but almost not quite, and it's called Neokinix. There's a slight difference between being paedomorphic and neotenic, but for our purposes, we can use them interchangeably, basically just meaning that the adults still look like the babies. These guys are really big. This salamander is this family. The Chinese giant salamander I showed you at the beginning of lecture is in the same family. They are found in Ohio. They have this very wrinkled flesh that increases surface area for gas exchange. And they're commonly called here hellbenders. I don't have any idea where that name comes from, but it's awesome. And this is, so the species we have here in Ohio is Cryptobranchus allegheniensis, or the eastern hellbender. I've never seen one of these in the wild. I would lose my marbles if I did, and hopefully that day will come soon. That's all I'm going to say about salamander diversity for right now. What questions do you have for me? All right, let's skip frogs. They're small and slimy anyway. And oh no, I thought I had a little slide with my right. What time is it? We're at 3:04. Yep. Yep. Which one do you want? The Cryptobranchus? Yeah, let me pull that up. You know what we'll do? Let's take a 5. Let's take 5. Take our mid-class break, and I'll come back and talk about frogs afterwards. And I will, you know, what I will do in the meantime is I'll find a nice image of nasolabial grooves and I'll pull it up in here for you. Sound good? So what time is it? It's 3:04. So let's start class again in 5 minutes. Take a stretch break. Get up out of your seats. Let's go ahead and get back to where we left off. So nasolabial grooves, you can see a nice diagram here of a salamander from a lateral view. You can see the costal grooves, and notice the nasolabial groove is this little basically groove between the nostril and the mouth. You can see, I don't know if you can see it very well here, but you can see it right here. It's more prominent often in males than females, and it often serves as like a channel for pheromones that help that are involved with courtship. And you can see a nice picture here. Do you remember the name of the species, Princeton? Western redback, yeah. The Western redback salamander, so you can see really nice big grooves here between its nose and its mouth. So imagine you had a little channel that would drip down from your nostril right to your mouth. It'd be probably pretty gross actually. But not for these guys. It helps them find and secure mates and reproduce. Most salamanders don't have this, Plethodons do. Sound good? All right, let's talk about frogs and toads. These guys exhibit a huge diversity of colors and shapes. You see some examples here. They're wild. Generally, with very, very, very few exceptions, they do not have tails as adults, and the few exceptions that do have very short tails. They have very long legs and feet relative to their bodies generally. Very often have these webbing between their toes or these enlarged toe pads. Extremely diverse reproductive modes, meaning that we have frogs that mate in water. We have frogs that mate in land. We have frogs where the dad pastes all the eggs on his back and carries them around until they hatch into little tadpoles. We have, check this out, a species of frog where they lay the eggs and then the dad comes along and swallows the eggs, but his digestive system has been completely modified. So his stomach no longer contains digestive acids and instead acts as a brooding pouch until all those little tadpoles hatch into little froglets, and then he barfs them all up. I'm not making this up, it's wild. There are species of frog where the eggs are laid on the back of the male, and they essentially, the dermis of the skin grows up around the eggs. So essentially the dad has a whole bunch of egg zits on his back. He carries them around and then they hatch and then they pop out of his back, which if you're into that kind of thing is really pretty cool. Frogs and toads exhibit metamorphic development, meaning that they have a larval stage, commonly called a tadpole or a polywog. And I have that here. So all kinds of cool stuff going on with frogs. We are not even going to scratch the surface of the diversity of frog reproductive modes and ecology in this class. We're just going to kind of give a broad overview. Here in Ohio, there's 4 families. We have Hylidae, Rhonidae, Bufonidae, and Scaphiopotidae. Those are the 4 families that we'll cover of frogs and toads here in Ohio. SK, go ahead. Oh, kidding. There we go. So before we get into that, oh my gosh, this one. But we're going to talk about all those right now. All right, so inurin anatomy. Before we get into it, some important things that will help us identify the different species. The first is a parotid gland, which is this kind of big kidney-shaped gland. There's 2 of them, generally just posterior to the eyes. Also, many exhibit what's called the postorbital crest, which is this kind of ridge between the eye and the parotid gland. They have the tympanic membrane, which is their ear. And if you've seen bullfrogs, you see their big old tympanic membrane, just kind of lateral and a little bit inferior to the eye. We have something called the dorsolateral fold, which is this line that goes generally from the back of the head all the way down to where the leg meets the body. It's like a fold of skin. Some species have it and some don't, and that'll be a defining characteristic. So diagram here that has some of the major parts of frog anatomy that we'll be referring to. Also notice snout vent length from cloaca to nose here in the frog. It's a little bit easier because they don't have a tail anyway, so it's basically the length of the whole animal. So let's talk about these guys in Ohio. The first family is Hylidae. These are generally referred to as tree frogs, although not all of them live in trees, although many do. The example I have here is Acris crepitans. This is the eastern cricket frog. These guys sound like this. This is an extended clip. Yeah. Oh, this is a really long. I gotta play the whole thing. And so frogs are unlike any other amphibians, rely on auditory cues, primarily for reproduction, but also some other aspects. I'll just say primarily for reproduction. And frogs exhibit distinct calls, and you can identify them by those calls. And later in the semester, we'll be learning the calls of about 12 or 15 Ohio frogs and toads and memorizing them. We're going to wait till later in the semester to do that, when we can actually have the treat of hearing some of them calling outside, which will probably be in about another 2 months or so. But you'll get to know all of the calls, or not all of them, many of the calls of native Ohio frogs and toads. Sid, let me get you in 1 second. These guys, the cricket frog has this kind of bumpy skin that looks superficially similar to what we call a toad, but note that it doesn't have this parotid gland. These guys are tree frogs, so they're not usually living in a lot of aquatic habitats, so they don't have webbing between their toes, but they do have these rounded terminal discs on toes. In other words, their toes end with these big kind of circular pads, right? So if we drew the frog foot, you would see its toes ending with these big, like open circular pads to aid in climbing when they live in trees. Their pupil is horizontally elliptical. That's really not that important. That's if you got one really close up and you're trying to ID it. Don't worry about that for right now. And they generally have longer and thinner bodies compared to a lot of other species. Sid, go ahead. Yep. And remember, all the lectures are posted, so you can just grab a screenshot. I mean, you can do it now, but it's easier, I think, to just grab a screenshot off of the video later. Cool. Let's talk about Ronidae. So these are often called true frogs. This is what, when you like imagine Kermit, he's in this family. Okay. The example I have here is a northern green frog, the Lithobates clamitans. You can hear what they sound like here. Whoop. Oh, wait. Way easier if I knew how to use a computer. Or if my computer worked. Well, let me talk while that's doing its thing. The adults primarily live at least close to water, if not in water. So like leopard frogs, bullfrogs, pickerel frogs are all in this family. So they have webbing between their toes, they have kind of a long and thin body. They often have a very large tympanic membrane. This guy in particular, look at the size of that sucker. That's huge, bigger than his eyeball. And many have these dorsolateral folds, and specifically the green frog has it, but the bullfrog doesn't. So that's a good way to tell a green frog from a bullfrog, is the presence or absence of this dorsolateral fold. Okay, computer, and the whole thing just crashed. That's neat. Well, I guess, Princeton, you're going to have to imitate the sound of a green frog. See, it's the only option here.
Well, to me, it sounds like someone like hawking up a loogie. And I don't know if I want to do that.
Yeah, but we want you to do that.
In front of everyone, so.
All right. Remember all that cool stuff we learned earlier today? That was really awesome. All right, I'm going to try this again, and I'm probably going to crash my computer again. There we go.
See, doesn't it sound like coffee, right?
It kind of does sound like coffee. I never really thought about it like that.
And that's all you remember.
I just thought it sounded like a giant rubber band being like plucked. Boom, boom. And so you'll hear these guys. I've seen green frogs actually here on campus even. So if we're lucky, we'll hear some this spring. That does sound like, yeah. Next family is Bufonidae. These are what's commonly called toads. Now I want to remind you, frog and toad as common terms for these anurans don't match up with their evolutionary history, right? So we call them toads, but just remember that there's not a good evolutionary distinction between frogs and toads. These guys have often what's called like warty skin. They have this parotid gland. They don't have that fold. They have these crests. You can't really see in this picture on the back of their head. And here you see the American toad. These are pretty common around here, along with Fowler's toads, Anaxyrus americanus. These guys are actually mating on the cover to my grandmother's pool some years ago. And this is what they sound like. You almost certainly have heard these around. I hear them every spring, kind of all over town. Sounds like a little spaceship from the Jetsons or something. You can actually hear some peepers in the background there too. I never noticed that before. Go ahead, Sid.
I don't know how smart of a question this is,
We'll find out.
but to produce that noise, like the constant, like, I don't even know how to describe it. Do they keep like moving their throat to make that, or is it just like a constant stream of one breath?
So that is actually like each kind of duration is like a single breath doing that, I think. Wait. I think that's the case, but I'm not 100% sure.
So
That's
they
a good
don't, like,
question.
chirp like most other frogs do in that sense, where it's like ribbit, ribbit, ribbit. It's just like one long ribbit.
Kind of, yeah. Mhm. Oh, and I want to fix one thing here. Do I want to go with? Sorry, let me put this up. Yep, I need to fix one thing real quick here. I have a small update on the taxonomy of one of these groups, and I didn't update this slide, and I want to just do it real quick, so I'm not putting up the wrong thing. Cool. All right, the last family of frogs and toads we'll talk about is Scephiopodidae, which is fun to say. These are commonly called spade foot toads. Again, they're toads, but they're actually not particularly closely related to the Bufonidae, the other toads. These guys are kind of smoother skin compared to Bufonidae, and they have this webbing between their toes, but they have this tubercle, this little kind of keratinized appendage on their rear feet. And so they're really good at digging, and they often live in pretty arid environments. And so they'll dig down during the dry season underground, stay nice and cozy, and then when it rains, they'll pop up in great numbers. We do find them in Ohio. So this is the eastern spade foot toad, Scephiopus holbrookii. And let me play what this guy sounds like. Good times. That's what they sound like. So they'll pop up after rain and be pretty prominent. I've never actually seen one of these guys. I'd love to do that. They're not around here in Ohio. They're further south generally, if I'm not mistaken. And that is the Anurans of Ohio, 4 families. Yep.
So does the Scaphio and Scaphiopodae spade?
Does it? I don't know what the root is.
Oh, it's what, so I assume.
Spade, that would make total sense. Can you look it up? What's Greek for spade, or Latin? Probably Greek. Scephio. Yeah, that'd be Greek. I never actually thought of that.
I mean, it would make sense, right? Because the spade is like a garden.
It would make perfect sense if that was the case. Yeah. I mean, it's certainly why they're called spadefoot toads. I just don't know if the family and genus name translate to that, but I'd be surprised if it didn't now that you say that. Chloe, go ahead.
You Explain what that last part was.
The tubercle? Yeah, better than that. Let's just,
It doesn't spade.
it doesn't, what does it mean?
It means the navicular bone of the tarsus.
Tarsus?
So because it's on the hind tarsus.
Oh, it's on the tarsus, so, okay, okay,
Tarsus foot.
that would make sense too. I'm trying to find a good. Oh, here we go. Look at this.
The word lamp.
All right, looks like I was way off. Okay, so spade foot. So it's got these little like keratinized or hard little appendages coming out of its foot there that allows it to dig, and it digs backwards, like it'll sit like this, and then boom, it'll just kind of back itself into the ground. And as I mentioned, they live in a lot of more arid environments, so they kind of, it helps them dig in this kind of drier substrate. Although what they show here is pretty wet. Does that answer your question, Clay? Yeah, I should. That's good. I got this slide in there now, so we'll have that for posterity. Sounding good? No, 2 questions. Yep, go ahead, Sam.
So the tarsus is
Oh. I said Sam, but that's fine, Sid. So this tubercle is like this little bony thing here, not the webbing between the toes. It's like a little protrusion that's hard, and it helps them just kind of dig a little more. Sid?
Sorry. Okay, so the tarsus, that's like on the front of your foot. Right?
Yep.
Of that. But
So your tarsal bones are your ankle bones.
yeah.
Mhm.
But so that's the same the front of their foot. I can't really tell.
Oh, the way this is, no, it's actually kind of on the inferior, so it's on the bottom of their foot. So it's like on the ground and helps them dig. All right, let's talk about Sicilians or Gymnophiona really quickly. I showed you a little bit about these guys on Monday. They're elongated, they're terrestrial, they can be aquatic but they often live underground. They have these annular bodies, meaning there's rings, kind of like an earthworm. They do not have limbs. They often don't have eyes either, or their eyes are kind of scaled over, so ineffective. And they do practice internal fertilization, but what's cool is they don't have a penis. They have an organ called a phyllodeum that's an outgrowth of some other tissue, and they can the sperm is transferred to the egg via internal fertilization via this cool organ called the phyllodeum. And the number of species in Ohio is 0. They're exclusively live in the tropics and subtropics around the world. You can find them and all of the equatorial region around the globe. And that's all I'm really going to say about them. And I think I'll come back to this in a second. I just want to see something real quick here.
It's Gymnophiona, right? With an M.
Oh yeah, it's misspelled there, isn't it? Ah. Thanks, Britson. What time do we got? That time? Okay. Okay. Okay, here's what I want to do is pause there, see what questions you have. Not saying a whole lot about Sicilians. We'll come back to them a little bit later in the semester. But honestly, we're not going to talk about them too much in this class. I'd love to. Cool. Yeah. Have you seen in the wild? Have you seen it? Or in the zoo? Yeah. They're neat. They're cool looking animals. So I mentioned that these guys reproduce via internal fertilization. That's in opposition to external fertilization, which is what our anurans do. And they do this by getting themselves in a position called amplexus. Amplexus is the mating position without an intromittent organ, and so there's external fertilization. In other words, the male and the females get their cloacas really close to each other, and the female lets out eggs at the same time the male lets out sperm. The sperm and the eggs find each other in the body of water where they're hanging out, and they meet, and if everything goes well, they the eggs are fertilized, and we have fertilized eggs that develop into tadpoles and more frogs, and the process continues. Just want to show you a little bit of video of this. It can actually be pretty interesting because males will sometimes be super aggressive when they're seeking amplexus and mate with like other stuff that's not females of their species. Oftentimes too, the females can exude a kind of glue-like substance from their skin that helps the male stay where they need to. But then when the male and the female are done, the glue stays sticky, and the females will end up dragging the males around for a while. So it's this really interesting reproductive act that we don't see in a lot of other organisms outside of anurans. Males often have specialized hind feet that allow them to hang on to the female really tightly and to grasp her as they're in amplexus. See some examples there, and I have one more video of examples. Oh, 9. Here we go. Which way? You can hear the American toad call in the background here, and once it gets into focus, you can hear some spring peepers in the background too. You can see their position in the water. Notice that to do this, they both have to be in the water. You can see this trail of eggs here. That's the female letting out this long trail of eggs. And at the same time, the male is releasing sperm. Sometimes you can see it as kind of cloudy, but the sperm is microscopic. You can't see it. And so they have to be close together for that to happen. Otherwise, sperm will just spread out in the water and won't last very long. Hundreds, if not thousands, like a lot. And we'll see some egg masses this spring. It's pretty cool.
How long? So, like, can they do it every day? Does it take like a long time?
For the female to produce more eggs? That's a good question. I don't know the answer.
For like chickens, it's like a day.
Yeah.
Other animals take a really long time.
Yeah, these guys, they only produce like 1 clutch a year.
Okay.
I think, let me double check that. It might be like 2. They might be able to wait a couple weeks and produce another one, but it's not like regular, like a chicken or anything like that. There are some lizard species that produce eggs regularly, like weekly almost. But these guys, it's just like a once a year thing. So amplexus, make sure you know that term. Do we have that down? That is the position, the mating position for anurans related to external fertilization. Zena, go ahead.
Is there any competition with that? Like Roger are in Texas? Are there other workers?
Oh yeah, you'll see like 3 or 4 males around a female sometimes. They're kind of scrambling to climb on top of her. And you'll see like a lone male who found like a really nice looking rock over in the corner of the pond sometime. Yeah, poor guy. It's kind of mayhem. And what's cool is when they're in that mode is they don't give a crap about anything else. And so you can go up there, you can like walk up to them and like poke them and they're just like completely oblivious to anything else in the world except for like mating and reproducing. So it's a neat time because you'll see a whole bunch of them in a pond and they're just there and you can go up and like check them out and take videos like that or whatever. So, yeah, but it's definitely competition. They both are. They're just on it. They got one thing on their mind, and that's to make baby frogs.
So, then if they're just completely like focused on that, are there predators that have time their like hunting with that particular breeder and dog?
Yeah, so it's complicated. In some species, that's true. In some species of, there's just some studies out. There's some frogs that call, in bats, cue in on the mating call of the frogs, and the bats are like, ooh, that's a frog, and they'll go and tag the frog. And so there's like this competition between frogs. They want to call in a certain way to attract mates, but if they do, the bats come, and there's all this stuff going on. With American toads, not as much. There's not a lot that eats these guys as adults, maybe raccoons and some other things, but they because they have a lot of toxins in their glands, like as adults. There's some snakes that will eat them too, but they mate at a time of year when it's colder and it's at night, so the snakes that are their primary predators aren't terribly active then anyway. So it can be an issue for sure in some species. For the American toad, it's not as big of a deal just because of the timing. Yeah, the thing about what else is going to eat those guys as adults. Yeah, some fish might come up, but in the wetlands where they reproduce are often ephemeral wetlands or small ponds where you're not getting anything big that's gonna tag a toad or an adult toad anyway. Little toads will get gobbled up quick, but not so much the big ones. All right, let's talk about your quiz, which is, what time is it? Oh, yeah, we have just enough time to work through this. So you have a quiz next week, not next Monday, but the week after, the 31st of January. I'm just giving you this information now, so you have it in advance. It's going to be a combination of short answer and multiple choice and drawing questions about everything we cover in the first couple weeks of class. You'll work individually on this, so it's not a group project, and it will cover material for the first 4 lectures. So that was the Wednesday last week, Monday today, or earlier this week, Wednesday today, and then Monday next week. I'll cover material on there. You'll have 20 minutes in class. My suggestion is make sure you look at the learning outcomes at the end of, I said lecture slides. This is actually should be at the end of every class program. Those are your study guide. That if you put all those on a sheet and then you can answer those, you can make sure you understand those learning outcomes, you're going to do great on the quiz. All of the questions will be based around those learning outcomes. So make sure you've got that down. It will cover phylogenies that we talked about. It will also cover a little bit of amphibian diversity, as well as amphibians specific to Ohio, what we covered today. And then we'll also ask you a little bit about mud puppy stuff that we'll talk about on Monday. Go ahead, Chloe.
When you say drawing, you mean like us drawing the phylogeny type of thing, or like us drawing the animals and labeling things?
Good question. It could be either. I won't judge you on your artistic talents or anything like that, but I might say draw a diagram of a salamander and point out the costal grooves, right? You don't need to make a beautiful looking salamander. If I did that, I would draw like a weird misshapen salamander, but you need to just be able to show me that you know where costal grooves are. Does that answer your question, Chloe?
Yeah.
Yeah, that's a good question. Thanks for clarifying that. And I will ask you to draw almost certainly ask you to draw a phylogeny of something. All right, we took our stretch break already. So I want to transition here a little bit and make sure, let's see. Your program for Monday includes, let me just pull it up and I'll just show you. I just want to walk through this real quick and make sure you've got this. We're going to have a Mud Puppy Monday on Monday next week. And to do that, here's your program for Monday. You're going to read a couple things on mud puppies. The one is a chapter from the Amphibians of Ohio book on mud puppies. It's just a general overview of their natural history. And there's some questions to keep in mind that we'll talk about Monday. But also we're going to read our first primary literature, our first scientific paper of the semester. And it's about mud puppy abundance in a river in Vermont and how a whole bunch of them died after they applied some chemicals to the river. You'll read about it. And what I want to make sure that you take away from this is I want to make sure that I'm explicit about what to expect in reading a research paper, because you don't read a research paper the same way you'll read a chapter or an essay or something like that. It's a very different kind of approach. And to facilitate that, you'll notice here, I say to do this, use the How to Read a Research Paper for the Big Ideas guide in Blackboard. And let me pull that up for you so I can just kind of walk you through that a little bit. Do that. There we go. And so this guide is in Blackboard as well. And so this just kind of has some tips for how to approach reading a research paper. And the takeaway message from this is you don't need to understand every little thing. Don't get hung up on the statistical methods, right? Especially the paper you're going to read next week. There's a whole bunch of stats about estimating population abundance and densities that I don't quite understand. I kind of know the general gist of it, but I don't understand all the details, but it's not important. I can still get the takeaway message from the paper, right? And so here's my recommendations for how to do it. I'm not going to walk through this in detail now because you have access to it in Blackboard. I'm just going to pull out a couple of things that I think are especially important. The first is just that if you're not familiar with what the paper is about, do a search and look at some images. I love to have the image of what the heck I'm reading about in front of me when I'm reading about an organism. It just helps me kind of visualize everything. I have just tips here for reading the title and the abstract carefully, and how you don't have to understand everything in the paper. One thing you do want to take some time to understand is the figures. The figures are really important. They're going to show you the main gist of the paper. So make sure you take some time with those and make sure you understand what the X and Y axes represent and what the takeaway message is. On the flip side, tables usually don't do a whole hell of a lot to help you understand the paper. In all honesty, it's a whole bunch of details that might be important if you're trying to look at one certain aspect of the study. But for the most part, I kind of gloss over tables in all honesty. So keep that in mind. And they can be intimidating. They could be like half a page of all these numbers. And you're like, oh, crap, that's a lot of numbers. Probably not that important to understanding the paper. Couple places here to note is the last paragraph of the introduction. So right before the methods. Usually that's a really nice paragraph to sum up what their hypothesis is and what they're testing with their study. At the same token, the first paragraph of the discussion is often a really nice summary of what they learned in the experiment. So if you read nothing else, reading these two paragraphs is really going to give you the gist of the paper. And so take a look at this in a little more detail and use that as a guide as you read this first paper. I don't want reading the primary literature to become like a really intimidating and a chore. And I think the best way to approach it is to understand that you don't need to understand every single detail. I read this paper yesterday and I didn't, there's again, parts of the stats, like I don't know how to do some of those marker capture things that they're talking about, but I don't need to because they're telling me what the result is, right? And that's what's important. We want to learn about the biology of these organisms and how we can get that biology out of these peer-reviewed papers. So check that out. And then I also have a quick survey on the program for Monday. It's just a quick question about which of the Bun Puppy field trips you're going to go on so we can plan accordingly. Details on the trip and then your outcomes. I don't have a reminder about the exam on here, but or excuse me, the quiz, but I'll make sure I put that on the next next program for you. And then a funny cartoon at the bottom there. And note that this program actually bled over into 2 pages. So sound good? What questions do you have about that? Okay, we got Mud Puppy Week next week. Just some reminders about the times. This is also on your program. We're going to check these guys out. And then let's see your program for Mud Puppy stuff. We didn't get to the video today. That's fine. We'll see if we get to it next week. We'll figure it out. That's all good. Let me know if you have questions between now and Monday. Otherwise, have a great rest of your week and weekend and all that good stuff. Thanks, everybody.